So today we will continue to talk about genetic terms, and first we will start with linkage disequilibrium. To understand what linkage disequilibrium is, you have to know what linkage equilibrium is. So linkage equilibrium is basically Mendelian law, right? Which refers to alleles that are independent from each other. They will inherit independently, and the expected frequency of alleles and the actual inherited frequency is the same, right? On the other hand, linkage disequilibrium is where alleles are dependent from each other, two alleles are dependent from each other, and therefore they will inherit at the same time and cause disequilibrium of expected frequency. Example here, you can see that from these parents, like they are the same, we expect to have equal amount of different children, right? 25%, this, and this, equally 25%. But if there is a linkage between two alleles, they will inherit at the same time and cause disequilibrium of expected frequency. So we have 45% of these, 5% of these, 5% of these, and 45% of these, right? It occurs because of recombination. So next we will talk about mosaicism. Mosaicism, we already talked about it yesterday, is presence of genetically distinct cell lines in the same individual, right? We have somatic and germline gonadal mosaicism. When we talk about somatic mosaicism, we talk about cells in our bodies. For example, somatic mosaicism, you have a girl, right, with features of Down syndrome, but you're not sure if she have it. And then you do genetic evaluation and you see that certain cells have pathologic 21 chromosome and certain cells are normal. This is somatic mosaicism. So in one person you have some cells pathologic, some cells normal. On the other hand, germline mosaicism is when you have father with pathologic gonadal cells, with pathologic it doesn't matter father or mother, but for example father, right, with pathologic sperm cells and normal sperm cells. So he will have healthy children and unhealthy children as a result. This is about mosaicism. Next we talk about locus heterogeneity. Is when multiple genomic loci undergo mutations, right? Multiple genomic loci. This is important point here because you can confuse it with allelic heterogeneity, where mutation occurs in one loci, different mutations in one loci, whereas locus heterogeneity, right? Locus heterogeneity, multiple genomic loci undergo mutations, and but we have the same phenotype. So we have multiple genes undergoing mutations, but the same disease occurs. For example, albinism or retinitis pigmentosa, right, have different gene that is associated with the disease. Okay, and next we go to allelic heterogeneity, so different mutation in the same locus, right, gene, result in the same disease in the same locus. This is the key point here. And allelic heterogeneity is very common for Mendelian traits. So that means all the disease associated with this is seen in allelic heterogeneity, right? So for example, Huntington disease, autosomal dominant, right? Rett syndrome, X-linked dominant, X-linked recessive, color blindness, hemophilia, sickle cell anemia, autosomal recessive, phenylketonuria, thalassemia, especially thalassemia, cystic fibrosis also associated with this, and Tay-Sachs disease. It is Mendelian disorders. Okay. Next is heteroplasmy. So heteroplasmy is presence of both normal and mutated mitochondrial DNA, resulting in variable expression in mitochondrially inherited disease passed from mother to all children. The point here is passed from mother to all children, right? So you will have pedigree of mother and father, right? And if mother have the disease, all of their children will have the disease, right? They will be all sick. But if her son will have children, they all will be healthy, right? On the other hand, if her daughter will have children, all of her children will be sick. This is mitochondrial. Answer in this case is heteroplasmy, right? This is important here. Then we go to uniparental disomy. Is basically offspring receives two copies of a chromosome from one parent, right? So it is true for Prader-Willi or Angelman syndrome. When we talk about Prader-Willi and Angelman syndrome, we say that Angel is for mom and Prader-Willi is for pop, father, right? And when we say that, we mean deletion, because deletion of mother chromosome gene on her chromosome will cause Angelman syndrome, whereas deletion in father chromosome will cause Prader-Willi. But when we have chromosome passed only from mother but not from father, uniparental disomy, we will not have father chromosomes, right? Only mother chromosomes are present, only from mom. And it basically means we deleted father chromosomes. And this child will develop Prader-Willi syndrome. If you get it, you have to digest it and understand, because a question will ask what's the problem here, what the pathophysiology here, and you can get confused when you see answer choices that are similar in your world especially. So if they talk about uniparental disomy and child have Prader-Willi, it is because he have two mother chromosomes. If they talk about Angelman syndrome and ask about uniparental disomy, it's because uniparental disomy, both alleles, both chromosomes are from father, uniparental disomy from father. Therefore, she developed Angelman syndrome. So here we see a question. A family has many individuals with phenotype characterized by hyperpigmented macros, axillary and groin freckles, iris hamartomas, fair chromocytoma, and optic glioma. What's the diagnosis? Neurofibromatosis, right? So, in our head, genetic analysis of chromosome 17 is performed because neurofibromatosis associated with NF1 gene mutation in chromosome 17, right? So patient 3-3, like this patient, and 4-4, like this patient. In the pedigree, like we see that these patients have the disease, right? But pedigree bear in the pedigree bear disease phenotype like they phenotypically present with a disease, right? But have an observed wild type locus on the examined allele. So we look at the 17 chromosome and we see that this chromosome is normal. Wild type means normal, previous to mutation, right? Why that occurred? It because of recombination, right? Because of C. What occurred? It just changed its location, but it's still there, and therefore it will cause the disease. So answer C, recombination, is right here. Next question here, a 29-year-old woman at 20 weeks gestation has an autosomal dominant disorder with 60% penetrance, right? A single nucleotide polymorphism marker is identified on the disease allele with a recombination fraction of 20%. Allele A and B for this single nucleotide polymorphism marker are indicated on the pedigree, this one, right? So question asks, which of the following is the probability the fetus will demonstrate disease phenotype, right? Phenotypically demonstrated. So we see that 60% here, right? 60%. 60% of him having phenotypical disease because it disease penetrance. Penetrance means will demonstrate it phenotypically. And 20% is that recombination will occur. So disease will be lost. And therefore 80% left that he will get phenotypical disease. What we do, we multiply 80% to 60% of penetrance, and we will get 4,800, right? And we will translate it into percentage, right? Divide it into 100 and get 48%. And the answer here will be E, right? 48%. That's how I solve this question. Next question is, a 50-year-old boy has a 4-month history of progressive shortness of breath with exertion muscle weakness and worsening exercise tolerance. Vitals are within normal limits. Physical examination shows bilateral ptosis. Muscle strength is low in all extremities. A pedigree is shown. The patient is represented by 310. So this is the patient. Question asks, which of the following best explains the combination of findings in this patient? So what we see here, we see that something occurred with his muscles, right? He had something with mitochondria, was suspected. This is first point. Second is that his mother passes the disease to all of her children. Her sons not passing disease, right? Even if they have it. Her daughters, on the other hand, passes the disease. Therefore, the answer here is E, right? Heteroplasmy. We're talking about mitochondrial disorder, which is passed only from mother to her children, but not from father to his children, because mitochondrial genes located only in oocytes, right? Only from mother. So next question, a 7-year-old girl with happy personality and mental retardation undergoes karyotype analysis that does not reveal a deletion of the expected UBE3A gene on chromosome 15. So UBE3A gene on chromosome 15 is Angelman syndrome, right? And also she has happy personality and mental retardation. Question asks, which of the following findings via fluorescent in situ hybridization is most likely to confirm the diagnosis in this child, right? So we said that we don't have deletion. So deletion is not the answer here, and we don't have it in our answer choices. So Engelmann syndrome, not deletion, we automatically think about uniparental disomy, as I said before, right? So it is paternal uniparental disomy here. Why is paternal? Because both chromosomes are from father. Therefore, we don't have maternal UBE3A gene on chromosome 15, right? And therefore, like, it is like it being deleted. We developed Engelmann syndrome. It is first. Then about paternal imprinting. Paternal imprinting is physiological process that occurred, right? And therefore it is not our answer here. Like certain chromosomes in our body, like paternal or maternal, are imprinted, and therefore it is necessary to have one part from mother, one part from father, because in the case of chromosome 15, paternal chromosomes are always imprinted, and therefore we will develop disorder such as Angelman syndrome in this case, but it is not pathophysiology. It is physiological process. Pathophysiology of this disease is paternal uniparental disomy. So last we will talk about populational genetics, and you have to know about genetic drift and natural selection, right? Genetic drift occurs non-naturally because of some dramatic event, right? You have lessened your population number dramatically, right? And as a consequence, you have less variation from the original population. So genetic drift divided to bottleneck effect and founders effect. Bottleneck effect is basically because of some poaching or dramatic event in the nature, you have lessened your original population, right? And then they multiplied, right? And you have less variability. For example, originally you had equal amount of yellow and red dots, and then eventually you have more red dots than yellow dots, right? So you reduced genetic variation because of it. Founders effect is when like originally you have small amount new colony, right? That is started by a few members of the original population. So this small population will multiply, and they will have less genetic variation because they started with small population, right? Natural selection is when alleles that increase species fitness are more likely to be passed to offspring and vice versa. Natural selection, right? Fitness means viability, which increases your strengths and like induce your survival mode, right? This is natural selection. And features that are bad for you will not pass across your population. That is populational genetics, and that's it.